This video demonstrates the importance of protein synthesis and the relevance of translation and transcription in a living cell. The video displays the significance of point mutation and the effects it has on a person's quality of life. This video was produced by Abby Anderson. To understand what protein synthesis is and how it works, you must recognise how DNA, mRNA and tRNA relate. DNA is a double helix shape and is made up from two strands. It holds the genetic information and instructions to help living organisms grow and repair. DNA has four base pairs C, G, A and T. C binds with G and A binds with T, the same way that G will bind with C and T binds with A. It sounds pretty straightforward, right? It's a bit more complex than that. So DNA is in every living organism and it's what codes us. The DNA base pairs are held together by hydrogen bonds. These bonds are weak, so that they're easy to break for protein synthesis. DNA is found in the nucleus of a cell. Small sections of DNA are called genes, and these help to code characteristics such as eye colour, freckles, hair colour, and so much more. Most of these genes are inherited from your parents, where one chromosome is passed down from the male and the other is passed down from the female. Genes are arranged in small coiled strands called chromosomes. mRNA stands for messenger RNA and tRNA stands for transfer RNA. mRNA is a single-stranded RNA that plays a crucial role in protein synthesis. mRNA is made from a DNA template when the two strands of DNA are unzipped. During transcription, mRNA is to carry protein information from the DNA in the cell through the nuclear pores and into the cell cytoplasm. The mRNA binds to a ribosome, and this is where the tRNA plays its role. tRNA is a type of RNA molecule that plays an important role in protein synthesis. When an amino acid is added to the chain, a specific tRNA pairs with the complementary sequence. This supports the protein being synthesized. If we think about it, it's like an assistant bringing the right ingredients to a chef so that they can make the perfect cake. But instead of ingredients, it's amino acids to make proteins. Protein synthesis has two main steps, transcription and translation. This is a basic explanation of each. Transcription is the first step of protein synthesis and translation is the second. Transcription happens in the nucleus of a cell where a copy of DNA is formed to make mRNA. The hydrogen bonds break between the DNA bases, and this is where a DNA template strand is exposed. Translation takes place in the cytoplasm, at a ribosome, and the mRNA codons are read. So, as we know, transcription is the first step of protein synthesis, and it takes place in the nucleus. A section of DNA is copied into a form of mRNA. When the hydrogen bond breaks between the DNA bases, a template strand is exposed, right? But really, in the transcription phase, there's a promoter region that causes the start of the transcribing of a particular gene. The RNA polymerase's job is to bind with the promoter region and copy the gene in an upstream motion. But before RNA polymerase can travel upstream, it must reach the operator, which is the start codon. However, an enzyme called a repressor blocks the movement by binding to the operator. The repressor must bind to something other than the operator to allow RNA polymerase to pass through and copy the protein synthesis until it reaches the stop codon. Once the DNA has been read and copied, a template strand is made called mRNA. So, moving on to translation. Now mRNA has been made, it contains the instructions for making a particular protein. The codons in the RNA are bases read in 3. Each codon is used for a particular amino acid. The codon AUG acts as the start codon and the other 3 act as stop codons. The start and stop codons signal the start and end of protein construction. Once mRNA has left the nucleus, it then binds to a ribosome. For translation to begin, initiation happens, where the start codon begins translation. mRNA is read a codon at a time. tRNA uses its anticodons to attach to the RNA codons. This means it brings specific amino acids that are needed to make particular proteins. The amino acid chain gets longer, creating what's called a protein chain. And lastly, termination is when the finished polypeptide chain is released. A stop codon enters the ribosome and it separates the chain from the tRNA and is free into the cell. So now I'm going to talk about point mutations and how they happen, why they happen, and what the effects are. In DNA replication, it's rare for errors to happen, but it is possible. Point mutations consist of subtle changes in a DNA sequence where a single nucleotide is altered. It can lead to diverse biological outcomes and can be inherited by the next generations. There are three types of mutations: silent mutations, missense mutations, and nonsense mutations. Silent mutations are when a nucleotide does not affect the amino acid sequence but may affect mRNA stability causing changes. Missense mutations are when an amino acid is substituted for another amino acid, causing the structure and function of a protein to be different. And nonsense mutations are when a premature stop codon is added to the sequence, leading to early termination where the protein isn't fully developed. This causes life-changing genetic variability in diseases. An example of this is cystic fibrosis. Cystic fibrosis is an inherited disorder and different mutations can happen for this outcome. The main mutation found with cystic fibrosis is a deletion of 3 nucleotides, leading to the loss of an amino acid. This then causes the protein to be folded incorrectly. A few symptoms of cystic fibrosis are sticky thick mucus, difficulty breathing, and some causes of infertility and a shorter life expectancy. So expanding on point mutation examples, I'm going to explain cystic fibrosis deeper. So it's a rare genetic condition that is inherited. Treatment and medications can help to support a person with this condition. When an altered gene is inherited from both parents, symptoms are present. If only one parent passes an altered gene, their child will not have symptoms of the condition but will be a carrier and could pass this gene to later children. Cystic fibrosis is life-threatening, and although it has no cure, there are a range of treatments to support a patient with this condition. Cystic fibrosis is an example of a recessive disease, meaning a person must have mutations in both copies of the affected gene. This means it must be inherited by both parents to have symptoms. Cystic fibrosis is caused by mutations in the gene that produces the cystic fibrosis membrane conductance regular protein. A person that has one mutated gene and one normal gene can be a carrier, and this can be inherited. A mutation in the CRTF protein leads to a malfunction in its job or does not work at all, leading to buildup of mucus and complications in other organs. This is because salt and fluid flow within the body isn't regulated. What determines gender and its inheritance? The sex chromosomes are X and Y. X is bigger as it carries more instructions, and the Y being shorter but determines the sex. A male carries XY chromosomes, whereas a female carries XX chromosomes. During reproduction, a female can only pass down an X chromosome, whereas a male can pass either an X or Y, which is a female or a male chromosome. This means that the male determines the sex as they can pass both sex chromosomes rather than just the female chromosome. This means that the ratio of female to male is still 50-50. Sex-linked diseases are traits inherited by X or Y chromosomes. Men are more likely to be affected by this and will express symptoms. This is because they have one X chromosome and only one copy of the disease allele. Because women have 2 X chromosomes, if one is affected, the other one is being compensated, so they will be a carrier rather than have symptoms of the disease. Women with an infected X chromosome can pass it down to their daughter, although she will also be a carrier, whereas the son will be infected and experience symptoms. Chromosomal abnormalities usually happen when there were errors during cell division. These cause significant changes. A few examples are stillbirths, abortions, and intellectual disabilities. It is possible to inherit these abnormalities, however, most are not inherited and may be caused by changes in the number of chromosomes during reproduction. An example of this will be Turner syndrome. The role of chromosomal abnormalities are important in Turner syndrome as they lead to the characteristic features of the condition. Turner syndrome is when there is a partial or total loss of the second sex chromosome. The condition primarily affects females where the X chromosome has structural abnormalities or a complete loss of an X chromosome. A few symptoms of Turner syndrome consist of facial abnormalities, reproductive issues such as ovaries not fully developing, a distinctive appearance, breast development issues, and other health risks. Genetic screening is a specific testing that can identify changes in genes that can cause health problems. This can support patients to diagnose some rare health conditions. Genetic screening is really focused on a bigger population than just an individual person. The process of the screening is by taking a sample of either hair, blood, skin, or amniotic fluid. This sample is then sent for testing for changes in chromosomes, DNA, or proteins. The outcome of genetic screening can identify individuals that are at higher risk of health conditions and help to support them. A few reasons that genetic screening can be offered are by doctors who think you may have an underlying condition caused by your genes, or a family member who may also have a health condition caused by changes of genes. A genetic test can help to understand if a health problem can be inherited and help to support with the correct treatment for the condition. The application of DNA recombinant technology is used to combine DNA from a variety of species and create different genes with new functions. An example of this would be type 1 diabetes, when the treatment being insulin. Diabetes is when a person's pancreas does not produce enough insulin for the body. Diabetes is a life-threatening condition if not treated. Insulin is produced when a single section of DNA from a human, mainly the gene that produces insulin, is coded into a bacteria plasmid. This is called a vector and delivers the new coded DNA to a cell. This replicates insulin the pancreas must create, and scientists use this to inject into a diabetic patient. DNA recombinant technology can also be used to modify the things that humans eat. In agriculture, pest control is very important and harsh chemicals can kill crops and be harmful to humans. DNA recombinant technology is used to make crops produce a protein toxic to certain insects. This helps farming to become more sustainable. A few examples of the moral and ethical issues raised by DNA recombinant technology would be animal welfare. Animal welfare is highly affected, and this is unethical as animals are used for testing. This can harm them or even have long-term effects that aren't yet discovered. Another ethical issue is consent. Farmers use DNA technology for crops that they harvest for pest control, or can genetically change the characteristics of some foods. However, not many people are aware about the genetic mutations that happen. This then leads on to health and safety risks with the mutations within the food humans eat, as we are unsure of the long-term impacts that DNA recombinant technology has on humans. This can also relate to medications or new treatments made and injected into the body. A moral implication could be the impact on the future generations and the potential of creating modified organisms that could be harmful to the environment and creating further risks. This could be the changes of genetics in an organism to create things inhumane that may affect generations. There may also be impacts on identity and privacy. As research continues, new treatments will be used on people that are affected by certain health conditions. This means that people who have experienced treatments that have been successful may have their personal data exploited without their consent, leading to the loss of confidentiality. So overall, the pros and cons of DNA recombinant technology are synthetic medications such as insulin, which helps patients with diabetes or other medical conditions. Gene therapy supports patients with treating or can cure inherited diseases. Another pro would be better nutrition, such as golden rice, to have sufficient vitamins. And then the cons would be high cost of treatments, moral concerns such as creating life synthetically, and other health risks such as allergies or different side effects. Thank you for listening to my video, and I hope you enjoyed it.